Hi, this is Miss Litton, and this is AP Bio Unit 2 Essay Review, Part 2. So if you're interested in the earlier reviews that we did over membranes and enzymes, I encourage you to watch the previous YouTube video. All we have right now, we have two chapters left. Well, we have one chapter left that we haven't covered, cellular respiration, so we're going to wait to review that next week once we finish it. So the only one we can currently review that we haven't already done already is the question about photosynthesis. So let's go over this together. So, oh, you can't see it, sorry, because I froze it. I'm frozen, there we go. So a net balanced equation for photosynthesis is quite simple, but photosynthesis is quite a complex process. The first thing when I read something like that, I would make sure I knew how to write the equation for photosynthesis. Can you do that? Can you write the equation? Assist me in the process, please. How would I write this equation?
Okay, 6H2O. Yeah.
Okay, where do you think you should start though? Where would be a good place to start?
The
sun.
The sun.
So energy, right, from the sun. Okay, now what? 6 CO2, okay, let's do that. Plus
6H2O.
6H2O, good. Goes to what?
C6H12O6.
C6H12O6. Plus 6O2.
Good, perfect. Okay, and the other thing I would do is I would know when I wrote an equation like that what got oxidized and what got reduced. Do you know?
The CO2 got reduced.
Into what?
C6H12O.
Good. So I would be able to go, if I was writing photosynthesis, I would go, the CO2 was reduced, and then what else? And I would write this got oxidized. We good so far? Okay, then it says, explain the photochemical and biological processes involved in the fixation of carbon dioxide in the chloroplast in the plant. So when you say the fixation of carbon dioxide, what are you talking about? Which cycle are you talking about? Calvin cycle. Another name for it? Light independent. Another name for it? Yes. But in order to explain that, you have to also talk about the light reaction. Your explanation should include such topics as photophosphorylation and the Calvin-Benson cycle. Diagrams are useful, but must be a supplement to the writing. So you might want to start, if you were writing, remember this is an old school way of this is just a way to make sure you understand the content. The newer type parts are the Google Doc that I shared with you. If I was going to go at this one, I would probably first break it down by the light reaction or the light dependent reaction. And I would talk about the important components of that light reaction. What are some key things we would want to discuss when we talk about the light reaction?
Sunlight.
Okay, the importance of light for energy and what purpose does it serve?
Breaks the water.
It doesn't break the water. Enzyme Z actually does that, but I haven't taught you that. But what do we need the light?
To start photosynthesis.
What does it do specifically?
Energy.
For what?
Photosynthesis.
Here comes the sun traveling in waves. Particles of light are called photons, and you're exciting what? Excite the electrons. Okay, so that's the whole point of the light, is to get the electrons at a higher energy level. Okay, so tell me something else that you need in a light reaction. Water. Why do you need water? What's the importance of water?
It gives up the hydrogens, which are the electrons.
Okay, so it's a source for what? Source of electrons that are used to reduce what?
The carbon dioxide.
The NADP. What else? You were saying it too. Source of hydrogen ions for the proton gradient. Needed to generate what? What are we generating? ATP. So if we're gonna generate ATP, we're probably gonna need some membranes, right? So we like the chloroplast. It's an easier one to explain. I mean, it happens also in prokaryotes that don't have chloroplast, but we need this because we can compartmentalize with membranes. We can set up a hydrogen ion gradient, right ladies, across that membrane to concentrate hydrogen ions on one side. Remember, we concentrate them to the innermost section of the chloroplast inside those hollow, green hollow pancakes, the thylakoid membranes. And so we concentrate our hydrogen ions to the interior, so as they move back out through the ATP synthase complex, we can generate ATP, right? And ultimately, we reduce NADP with those electrons that power up and give us the ability to create that hydrogen ion gradient, yes? And so from that, we can synthesize ATP. So you're gonna wanna explain that out. Things you might wanna include is reaction centers, right? And structure of the chloroplast. Okay, and then B is in this case it's called the Calvin-Benson cycle, but you know it's the light independent reaction. Okay, and what are some things we need for the light independent reaction? Now I'm gonna start on a new page because it's getting busy. What?
Independent.
Oh, thank you. What do we need for the light? It's just like the notes. The light independent reaction. You can always think, what do you start out with and what do you produce on both of these reactions? That's a good way to write it. So what do we start out with in the light dependent reaction? Okay, CO2, good. And we're gonna do what to that? We're gonna reduce it into what? Good, okay, what else do we have? Hi, what else do we need to do the light independent reaction? ATP. We need the ATP to energize and form our G3P, or another name for that, old school is PGAL. What else do we need? We need reduced NADP as a source of electrons in order to reduce the CO2 into glucose, yes? Okay. We need a location. Where are we going to do this? In the stroma. Good. And we can talk about, when we look at, this is a hideous chloroplast. Okay, so our cycles are taking place out in the stroma, and then along the light reaction that's taking a place along the thylakoid membranes. So our ATP is generated out in the stroma, and that's where it's used. Our reduced NADP is generated out in the stroma, and that's where it gets used. Okay, now you could talk about the light independent reaction. You could talk about how we could do it in 2 batches of 3, right? Of 3 CO2. So we put 3 CO2s in, and we end up getting 1 3-carbon molecule out, and that's that G3P or PGAL. So by doing it 2 times, we have 2 halves of glucose, or 2 G3Ps. All right, and so you would want to address all of those issues. You want to be able to get it as succinctly as possible, make your point, but make sure you hit the important parts, like why you need each thing and what product you get from each item. Okay, so again, it says diagrams are useful, but it has to be a supplement to any writing that you're doing. You want to be able to convey it out. Then B says, describe a lab in which you can measure photosynthesis, include your setup, controls, variables, and results. That was the first thing you did for me this year, was a lab. What was that lab we did? What? We had a beaker. What did we put in our beaker? What? Leaf discs. Okay. And there were leaf discs. I agree with you. And then what else was in that beaker? Water. What else?
Carbon dioxide.
Carbon dioxide coming from what? CO2 from that bicarbonate baking soda, right? Good. What else was there? What else did we use? Oh, this is not working out. Okay. Let's do it like, okay, yeah. I'm trying to.
There you go. It's beautiful.
Good. Yeah, so glad we're recording this. Okay, so light, right? Okay, what was our dependent variable?
The amount of disc that flows.
Reduction of oxygen.
Okay, time and number of discs, right? And then from that, we can infer the amount of oxygen generated as a byproduct of photosynthesis. Okay, what was our independent variable that we did?
The amount of carbon dioxide in the water.
Boom. Okay, amount of CO2 in the water. And if we were going to write a research hypothesis, we would say if what? If I was doing a research hypothesis.
If we increase the
Amount.
amount of CO2,
Mhm.
then it'll increase the amount of disc that flows.
It'll increase what?
The
Amount of disc that floats per unit time, right? So when they ask you to design an experiment on an exam, you do not need to come up with a new and different one. Use experiments we've discussed or done, okay? And you want to just make sure that you have your independent variable, your dependent variable, and if it's a good experiment, it also has a control. You want to have a data that's quantifiable, that has a number, right? That you can use math on to support, to validate whether or not that it's statistically significant on the number of discs that rose. And just to remind you, those discs, and they would normally float, but what did we do to them so that they started off at the bottom? Because our start of our experiment was discs that were at the bottom of the cup. So how would we get them to start being at the bottom of the cup?
We suck the oxygen out of them using the spray.
Okay, we used a vacuum to remove the gas. It could be any kind of gases out of that, and infuse those discs with what? That water that we were using in the experiment, whatever percentage of sodium bicarbonate. Did I already say the control, what the control could be, or did you already tell me? What would be the control? Distilled water. Okay, good. So you created a vacuum using a syringe so that the discs were filled with water, and then whatever water solution you were using. And then the assumption is they would do more photosynthesis than what? Cellular respiration, right? Because if they were doing net, if it was the same, then they should stay at the same level in the water, right? But if they're doing more photosynthesis than cellular respiration, because we have the CO2 in solution, then we are assuming that's going to produce oxygen. Oxygen will take up the space in the leaf discs, and then they will float as they have increased amounts of oxygen produced. Okay? So that would be your experiment to back up your understanding of photosynthesis. Now, there's only one question on this one left that we haven't already covered, and that was it's cellular respiration, but we haven't talked through the steps of that, and then a lab you could do for cellular respiration. We're not doing a lab for cellular respiration, but I'll give you one you could do, for cellular respiration. And then in your, the ones that are more AP-like questions, there's another question in there we haven't addressed yet, but it's more along the lines of cellular respiration. So we'll address that one when we do cellular respiration there. Okay, any other questions on this? Okay, you're super smart, super proud of you, and hope you have a great rest of your day.